Across Europe, hundreds of traditional maize varieties are conserved in gene banks. These landraces represent centuries of farmer selection, local adaptation, and genetic diversity. Yet, until recently, their full potential for modern agriculture has remained largely unexplored. Maize was introduced to Europe in the late 15th century. Since then, farmers and breeders have developed a wide range of local varieties adapted to different climates, soils, and farming systems. These landraces may contain valuable traits for breeding crops that are resilient to climate change and environmental stress. To investigate this hidden diversity, the ECPGR European Evaluation Network, or EVA, was established as a public-private partnership. One of currently 6 crop networks, the EVA Maize Network brings together gene banks, research institutes, and breeding companies from across Europe. Their goal is to systematically evaluate maize genetic resources preserved in European collections. In this study, 626 maize landraces from 8 national collections were analysed using high-throughput genotyping. Because maize landraces are outcrossing and thus very heterogeneous populations, multiple plants were sampled for each landrace and their DNA pooled before performing SNP genotyping. The analysis then used a prediction model to determine the frequency of different alleles, applying a threshold for statistical assignment to different genetic groups. The genetic data grouped these landraces into 9 major clusters, reflecting their geographic origins and historical introductions into Europe. This genetic structure reveals how maize diversified after its arrival on the continent, and showed how breeding combined materials from different genetic groups. From this collection, 588 landraces were evaluated in multilocation field trials. These experiments were conducted across 11 European sites over 3 years. 5 commercial hybrid varieties were included as reference standards to compare data from different locations and years. Key agronomic traits, such as flowering time, plant height, and ear height, were recorded. The results revealed large variability among landraces for all measured traits. However, the phenotypic clusters only partially match the genetic groupings. This indicates that environmental adaptation and complex trait interactions play a major role in shaping maize performance. These findings demonstrate that European maize landraces represent a rich and underutilised resource for breeding. They offer genetic traits that can support the development of maize varieties that are better adapted to local environments, climate change, and sustainable agriculture. By combining genotyping with large-scale field evaluation, the EVA Maize Network provides a powerful framework for unlocking the value of gene bank collections. This approach strengthens food security and supports the long-term sustainability of European agriculture.